Hello students, in the last video we have discussed the whole process of fertilization and completed the process of fertilization. Today we would go to the next step or the next phase of human reproduction that comes under another topic. That is what it is known as post-fertilization events. As we know, fertilization is the main differentiating point between asexual and sexual reproduction. So, the processes or the phases that occurs before fertilizations comes under pre-fertilization events, that is gametogenesis and gamete transfer. And then fertilization occurs, and all the next phases that occurs after fertilization are termed under post-fertilization events. So, today we would discuss another point and another important phase that is known as cleavage. Now, in the last video we have done an overview on this, that when the fertilization occur at the site of fertilization, that is your ampulla region of your fallopian tube, and after this fertilization, now this is the site of fertilization. So in the isthmus part, this is the isthmus part of the fallopian tube. This isthmus part is actually responsible for the cleavage process. Now what is cleavage? So we would say this term was given by von Baer. So a scientist gave this term cleavage. It is relevant to cell division, but it is different type of cell division. So we would say once a zygote is formed, now it undergoes repeated mitotic division, and this is termed as cleavage. So what is important? The set of divisions that would occur after the formation of zygote. So once the zygote is formed, it will undergo consecutive mitotic division to increase the number of cells, and hence it is known as cleavage. Now why cleavage? Usually we know because of cell division the number of cells or the number of daughter cells increases, and due to increase in number of daughter cells the whole structure of that cell increases. But here, due to division, the cell size would not increase, it would keep on decreasing. That's why it is not known as normal division, it is known as cleavage. So we would draw a diagram here, that is A. So here we would write this A is what? It is the secondary oocyte, which is secondary oocyte, which is released from ovary. So once this secondary oocyte is released from the ovary, it will reach here, that is the ampulla part. So this is B. Now to this sperms would attach, and here only we would draw another diagram and we would write this. This represents the zygote. Now we can say once this zygote is formed, within 24 hours another structure, that is two-celled structure, is formed. So this we are showing A, B, and here we are showing this is C. Now, as we said, this will undergo repeated division. So, after this, again a structure is formed, like this. Again a structure is formed. So, we can show this by D and ultimately E. Until and unless it reaches to the end point of your fallopian tube. From here, the uterus starts. So, what we are saying, now these structures are produced due to repeated division in the cell or in the zygote, which is a diploid structure, and hence that process of division is known as cleavage. So we can know one thing, that cleavage or during this process, the daughter cells or the new cells which are produced is known as blastomeres. So there is a special term, that is what? Blastomeres. They are not known as daughter cells, they are known as blastomeres. So it is called cleavage. It is called so because the size of blastomeres keeps on decreasing until it is equal to the size of a somatic cell. Now, we said somatic cells are those cells which are responsible for the growth in our body. Once an organism is formed, first that organism, when it takes birth, it would undergo growth. For that growth, these somatic cells undergo mitotic division or equational division. So here, usually in a cell division, the number of cell increases, then the size of cell increases. But here the size would keep on decreasing until and unless it would be equal to the size of a somatic cell. That's why it is not known as normal mitotic division. A special term is given, that is what? Cleavage. Now, a question arises in your mind, why is it so that a process of cell division is decreasing the size of the overall cell? So, reason for this is, that is very important. Reason is, in this cleavage process, there is only synthesis of DNA. So we can say that is only S phase and then M phase. So they would not have the two main phases of the interphase or two main subphases of the interphase. As we know, during the cell division, we have interphase and then M phase or the division phase. This M phase is also known as division phase. So, interphase is further divided into G1, S, G2. So, here is only S and then M. S and then M. No G1 and G2 phase. That's why actually the overall size of a cell increases due to accumulation of more and more cytoplasm. And if there is no G1 and G2 phase, there is no accumulation of cytoplasm. Hence, the overall structure of the cell or the size of the cell would decrease and not increase. Now, here we have seen this A is your secondary oocyte. So, we would draw the B diagram. Imagine this is your secondary oocyte. That was an haploid structure. At the B, we would show this is your secondary oocyte, or we can say it is egg which is lined by zona pellucida, and above that there are follicular cells, and to this follicular cells we would show entry of what? Sperm, or attachment of sperm, and ultimately this would change into a structure that is called zygote. So we would say the pronuclei of sperm and egg has fused. So now this is ploid. It will have that zona pellucida intact to it, like this, and the few follicular cells above it. So this is the structure of zygote. Now this is formed here. This is the zygote part. So zygote is formed. Now this would undergo, now it undergoes mitotic division. So this transition from B to C occurs within 24 hours. So after fertilization, within 24 hours, the first mitotic division would occur in the zygote, and there would be formation of two-celled structure. This is two-celled structure, and we would say these are what? These are blastomeres. We said they are not called daughter cell, they are called blastomeres. So it is a two-celled structure with two blastomeres, and this is important. This is called plane of division. So we would say plane of division is from animal to vegetal, animal pole to vegetal pole. We discussed these two poles when we discussed the structure of egg. The pole, or that region of the egg where the nucleus was present, was known as animal pole, and opposite to it was a vegetal pole. So animal to vegetal pole, this axis of division occurs, and this is an holoblastic cleavage. We will discuss it. What is holoblastic cleavage? So this is holoblastic cleavage. Holoblastic cleavage is what? When the axis of division or the plane of division would pass all through the egg or the structure. It would pass throughout the structure. Then it is known as holoblastic cleavage. So here the two-celled structure is formed, and then this two-celled structure would convert to this. That is what? That is a four-celled structure. We would show like this. Four-cell structure. And ultimately to E part, that would have multiple number of cells, or that is called 8-celled structure. So first mitotic division is equal to 2-cell structure, second 4, third 8-cell structure, and ultimately when it would reach here to F part, it would be converted into mass of cell, that is 16-celled structure. So once it reaches into the uterus, this becomes 16 or 32 and more celled structure. Now we would show, this is the wall of the uterus here. So F, after F we would show this, that is G part, that is important. So the structures or the stages that occurs in this cleavage process is first structure which is formed just after zygote. We said first zygote is formed. just after zygote, a structure is formed which is called morula. Now this is the term given to 8 to 16 celled structure. So when after consecutive mitotic division there are multiple number of blastomeres or 8 to 16 celled blastomeres, they are also called solid ball-like structure which resembles mulberry ball. So it would appear to be a mulberry ball-like structure which has large number of cells which are compactly packed. There is no more space. So once this morula is formed at the F region, then after more division, this morula will change into blastula. It is also known as blastula or blastocyst. So there are three main terms given here. Zygote, that was a single celled structure. This is what? Single celled structure, but it was diploid in nature. Now that diploid cell, single cell, keeps on dividing by mitotic division to form 8 to 16 celled structure which is resembling like a solid ball, that is known as morula. But up till when it reaches to the uterus, it is converting into another structure, that is blastula. So we would write here G is what? G is another structure. This was solid ball, but the cells were not arranged. Here the cells would arrange themselves. So we can say together with the cell division, now what is occurring? Cell differentiation. We have discussed this in the cell chapter that cell division cannot occur alone. Cell division means increase in number of cells, but the cell division is always followed by cell differentiation, that is arrangement of the cells. So here the arrangement would occur where a layer of cells are arranged to the periphery which are smaller in size, and another cells are accumulated here in the central part. This is known as inner cell mass. The cells which are larger in size would get accumulated here, and this layer is known as trophoderm. So now the cells present in this structure would get arranged into two layers. The peripheral part is known as trophoderm and the inner one is known as inner cell mass. Now there is a cavity here. This one is the cavity. This is known as blastocoele. That's why the name is given blastula. So we would say 32 and more celled structure is called blastula. So once we have done with this cleavage, the next step is the process of implantation. But zygote is not implanted, morula is not implanted. What is actually implanted in the wall of the uterus? It is the blastula. So this structure is going to get embedded in the wall of the uterus. That's why the wall or the inner wall of the uterus must be rebuilt again and again during the menstrual cycle, because actually that implantation process, that is sowing. When we grow a plant, we sow the seeds. We sow that seed in the soil, then uprooting occurs. Similar is here. We are sowing that seed or sowing that blastula into the wall of your uterine or uterus. So this is the set of changes that occurs in the fallopian tube or the isthmus part of the fallopian tube, and hence it is known as cleavage, and these are the changes would occur during the cleavage. But this cleavage is very much important, so there is a classification of cleavage on two bases which we will discuss in the next videos. Thank you.